boys and girls. Here with Demeter and Trace. And we're going to go through a video by John Sutherland. I keep wanting to say Donald Sutherland because I like him too. So anyway, but it's not. But let's see, if you guys want to stop the video, you should say Dannykins loudly, because then I'll pause it. But I'm not sure how much. We could try to go through the whole thing. It's 2 hours long. 2 hours and 1 hour and 7 minutes long. But we could give it a shot. What do you think?
Well, I mean, you can skip the introduction part and just go deep in the chemistry part, for example.
Where would that be exactly? That would be probably at about. Oh, let's try starting it. Try starting it right here.
Yeah.
And let me try to get things lined up. Okay. First of all, I got to figure out whether or not the sound is coming through. Hold on a second here. Oh my goodness. Okay. What is wrong with that? I don't know what is wrong with that. Oh no. I did something bad. Very bad. Okay. Kind of got it together, I think. Try it.
And so people have tried to sort of imagine going down further by saying, well, what would be the minimal system you'd need to have a living system? And people are approaching this experimentally by knocking out redundant genes and so on in microorganisms. But just hypothetically, you kind of assume that you need informational molecules if you want to be capable of Darwinian evolution. You want to be able to have catalyzed either replication or synthesis, because in the absence of catalysis, those are going to be difficult, maybe achievable, but probably difficult. There's arguably no point in genetics or metabolism unless you have some sort of compartments. You've got to sort of keep the good stuff that you make away from your competitors. The best way of doing that is compartmentalization. And finally, you need building blocks and energy if you're going to be able to do anything. Now, imagining how you could get all of that together at the very outset is very difficult. And so pretty much everyone has agreed in the past that this puts such demands on hypothetical prebiotic chemistry. Surely you're going to need different chemistries to make the various different bits. Surely you need different chemistry to make amino acids and nucleotides and lipids. And surely those different chemistries are going to interfere with each other. These are assumptions, right? And so what people have done, they've all agreed that it looks too complicated to start the whole shooting match in one go. So they've said, let's start with one subcomponent, one aspect of the system, and imagine that could then give rise to the others later on in time. And the particular subsystem that people have selected to come first depends on their background, their intellectual bias, their subject discipline. So there are some who've said, genetics is everything. A nucleic acid, RNA must have come first. There are others who've said, no, proteins must have come first. And you get the gist here. There are still others who say, no, membranes must have come first. And finally, people who say, forget RNA, forget everything else. Metabolism's the be all and end all. So you've got to start with metabolism. So notice what you've got there is everyone agreed that it's apparently too difficult to make everything in one go. And then people are polarizing, in fact, quadrupolarizing. And 20, 30 years ago, you went to an Origin of Life conference in a room like this with four corners, and the people in the room would congregate or segregate into the four corners and trade insults with each other. RNA is more important, RNA is too complicated, metabolism is much simpler. And after listening to that for quite a few years, I started to think, well, one of the reasons this debate is so hard to sort of deal with is because all of these people are making good points. So is there a way of actually taking some of those good points that everyone's making and trying to put them together into a more cohesive picture? And we started to do that by thinking about whether or not it's appropriate to view biology as a modular system. So I've described the fact that you have subsystems of catalysis, the genome, compartmentalization, and so on. But to what extent are those independent modules? So if, for example, you look at nature's way of making RNA, it uses protein to make RNA. RNA polymerase is a protein. If you look at the way that nature makes protein, it uses an RNA machine to make protein. So protein and RNA are, yin and yang. They are not something which biology readily suggests could be separable in a modular fashion. If you look at membranes, membranes are not just composed of lipids, they're chock full of proteins. And some of those proteins play a big role in metabolism. So for example, the way in which the energy associated with a transmembrane proton gradient is harvested in the form of ATP synthesis involves a very complex membrane protein. So is it appropriate to separate metabolism from compartmentalization? It's conceptually difficult, perhaps, to do. And finally, if you look at metabolism is a couple of thousand reactions, a couple of thousand or so small molecules, and it's regulated. And biology regulates metabolism really carefully at the level of transcription, at the level of translation, and allosterically, once you've actually made proteins by translation. And if you don't regulate sort of metabolism, you end up with a mess, okay? So we started to think, there are some questions we've got to start addressing here. Were the conditions for prebiotic chemistry compatible with life? So do we have to make all the bits and pieces we need to assemble a functioning system?
I just want to point something out here that I read in a paper a few months back. Basically, if you have alpha helices in proteins, if they form helices, and they will gravitate to the membrane thermodynamically, and they will embed themselves in the membrane. So that's thermodynamically favorable. So alpha helices just ending up in the membrane is a possibility for prebiotic life. So,
I also want to stop a little bit here. I wanted a little bit before to stop, but anyway, so he presents some kind of classical prior 2000 style of thinking for origin of life.
Yep.
Where you have something first is going on. And also he points out that, of course, later on he will show that in fact you don't need to think in this particular way to come to say that something should first, but what actually is the bottom of the entire story. And also he pointed that, for example, if you go from top to the bottom to look for origin of life, he said that here you will come to all of these complicated interactions, metabolism, proteins, RNA, blah, blah. Yeah. However, I want to mention something that if you go from top to the bottom analysis, you will not stop where the LUCA is. You'll go much deeper before LUCA by this analysis, because you can analyze the domains of the proteins, which is the more ancient, the first domains that actually appeared. It's a very interesting paper from Gustavo Anoles, and he showed very clearly that which are the first domain, and he distributed in the way of the chronology, in a chronology way. So it's not true that you'll stop where the LUCA is if you go from top to the bottom, but you'll stop somewhere in the first domains. And also you can figure it out that some of the motifs of these proteins are actually the more ancient than the others. The same is true for the sequencing of RNA. For example, you can see that some pieces of RNA are more ancient than the other pieces of RNA. And we know that these pieces of RNA are part of the tRNA, a part of the ribosomal RNA, the small and the bigger subunits, and probably some ribozyme also at the most station. So the pathway from top to the bottom, it's not like having some complicated picture, but actually it goes very deep. Unfortunately, from the origin of life, where you have just a few peptides, short peptides, to the moment where you have a domain, organized domain, it's a huge leap between. It's a huge leap of evolution. So it's a very long time to come from the just short peptide to some kind of domains, protein domains.
Right. Yeah.
So anyway, so let's keep listening.
There's one thing I would throw in there too, because you're talking about going beyond LUCA with the limitations of phylogenetics and going beyond LUCA, and something that I've come across with respect to that is the work of Eric Smith and Morowitz, working on the logic of metabolism. And what they do is they use phylogenetics and metabolomics to check one another, going into like a deeper dive of reconstructing what the most ancient aspects of LUCA are. So you can take phylogenetics to the point of LUCA and then push even further by reconstructing what are the more ancient pathways in metabolism, which is something that they did. So there are, so I definitely agree with what I just heard, that there are ways to get beyond LUCA and the limitations of phylogenetics.
Yep.
Yeah, I mean, okay. Yeah. Okay, let's keep going.
Proceed.
Under conditions in which they could function in a living system? Or can we make some of them by chemistry that's not compatible with life, store them up, and then later use them to initiate a living system? Do we actually really need completely different chemistries to make the various subsystems of the cell? That's an assumption. People have just assumed you need different chemistries. It's not being rigorously or exhaustively experimentally tested. So we should start doing that. Because if so, would these different chemistries really have interfered with each other? And to what extent would you have to separate bits of chemistry to stop that interference taking place? Was the chemistry indiscriminate chemistry like the Miller-Urey chemistry, where you make not just traces of the amino acids that you as well, and it just so happens that you have, a few compounds that you want in a morass of other compounds that you don't want? Or was it an intrinsically favored chemistry leading to a restricted set of products that just happens to work? Okay. If you need to do what chemists are always doing in the lab, which is adding reagents sequentially, purifying things and partitioning things, how do you do that without guys or girls in white coats to do it for you? How does the environment allow that sort of thing to take place? Which components are ancient, which are more recent? And finally, could we actually get the whole thing assembled all in one go? So these are a set of questions we posed 10, 15 years ago and started trying to address. And
Danny,
let me frame. Yeah, Dries, go first.
Heard, thank you. Really quick, if you just go to Google Scholar and you type in continuous reaction network and probably throw an origin of life in there, you'll get a series of articles about exactly what he's talking about. Continuous reaction networks are basically, when he's talking there about how you want to have a series of sequential steps without human interference. The technical term that they're giving to that is a continuous reaction network, something that'll happen on its own without the researcher's interference. And there's a lot of work that's been done, a lot of papers that have been published finding continuous reaction networks. That's all I had. Demeter, you're up.
I wanted to point out, look, the beauty of science, exactly in this previous 5 minutes. All of these arguments that he pointed out was heavily overused from creationists, right? You cannot have this. You cannot have a white man a man with a white coat on the prebiotic earth and trying to put that. You cannot do this. You cannot do it. Look at here. You have the same argument, but these arguments are not like a stop. I cannot do science. I cannot discover. These arguments are valid questions for discovery, because yes, indeed, there is a problems here. We see all of that problems, but also you see that you're looking actually for discovery, for the next step, for something that will give you the nature answer. Answer will be given from the nature. It's very interesting to use, because if you look, all the creationists, they will look absolutely identical arguments, which he pointed out.
Yeah.
About that.
Yeah, it's a pretty common creationist argument that you have to have purified reagents and pristine conditions. And actually, if we'd had those, I don't think life would have happened. It was the fact that there was this chaos, and then there was a particularly favored reaction somewhere in that chaos that managed to punch its way through. And if I mean, we could go on for thousands and thousands of centuries trying to figure out, by doing one reaction at a time, right? Trying to figure out exactly what the pathway is, or try to make that pathway. Whereas life was a tremendous number of different compounds all together, and the ones that fit were the ones that ended up shining through. So, and as it turns out, like the literature I've seen in the last month or so, there is a lot of reason to believe that that the structures that we do find in life now are the ones that tend to be very common. We found them in meteors, surprisingly, with the ribose hooked up to the right spot on the nucleobase or the nucleoside, what is it? Nucleic acid, whatever. Yeah. Anyway, which is kind of surprising. It's surprising that we found such a high quantity coming in the right configuration. That had to be because that was what was likely to pop up. So anyway.
Yeah, and he will point it out. I want to comment. Let me finish this particular imaginary graph for what is life and aliveness. He pointed on a Y. He put aliveness at the time. So let's see this.
Fully alive.
Yeah, he's wrong.
Okay.
We view the origin of life as being a transition from a system which is not alive to one which is alive, proceeding in stages. So we don't like to think of a Frankenstein's monster moment where suddenly something that's inanimate is transformed into something animate. We think more in terms of some of the major attributes of a living system actually being able to function independently of each other and be acquired sequentially. So those big innovations to the system could be things, for example, like vesicle replication, nucleic acid replication, translation. Those could be things which, taken collectively, will constitute a living system, but taken individually, don't. And we see this as being something which is chemically deterministic. In other words, if you played the tape again, you get the same result, until you reach the point at which you're making macromolecules which have got sequences which are long enough that you're not going to be able to sample the same sequence space if you did replay the tape again. Biology tells us that you can have multiple solutions out there in sequence space to the same problem. So once you get to the chemical contingency part of this diagram, if you replayed the tape, you might well get the same function, but a different sequence bringing about that function.
We should have a name for that moment, basically, when the tape would not be played the same way. When you're sampling the sequence space, you would probabilistically end up with different life forms. I don't know.
That's an old question of in a new way formulated.
Yeah.
So, and here I don't like this figure from John Sutherland, particularly because it's kind of, it's not because it's not correct, but there is a moment that you have a boundary between chemistry and biology. And usually so far, most of the scientists think that you have to have some sort of gradual accumulations of interactions until the moment, aha, you have life.
Yeah.
However, that's not important because according to John Sutherland and many more, of course, if you, for example, have some process simulated in the environment, like let me say RNA replication, somehow it's happened RNA replication, that's not life. If you have just translation, that's not life. But if you have both translation and replication, now we already have in some sort of compartmentalization, now we have a life. That's not the way how we should think about origin of life, because you have a moment where you start to have Darwinian evolution. Before this moment, you don't have Darwinian evolution. You have just a bunch of chemical reactions, but though there's no information that will be harbored, that will be go from generation to generation. And that is the moment where you can call it life. And here is the how it's called, heresy for the modern biology, because you call by this definition, you call life something extremely simple, but it's capable for Darwinian evolution. Therefore, you cannot say fully alive, because there is no something which is less alive and more alive. You either have a system which is capable of Darwinian evolution, or you don't have this system which is capable of Darwinian evolution. And that's a crucial new moment, which I hope very soon everyone will understand.
Okay. Proceed.
Yeah.
And what I've drawn here on the bottom basically is what occupied the first 25 years of my career, the synthesis of building blocks by inherently favored prebiotic chemistry. We always had it in mind to try and make polymers at some point. We did not anticipate that making the monomers would take us so darn long. Okay, 25 years and still counting to finish it all off. And we recognize that contributes nothing to the aliveness of the system, which is, after 25 years of your career, is a rather humbling realization. You're working on the origin of life and have contributed nothing towards, making a system that's alive.
Dan Gens.
What about the conditions? Well,
yeah, so I think he's being way too hard on himself there. Obviously, if you have synthesized the vast majority of monomers and shown how that can be done, you have contributed something to origins of life research. Granted, there's a shit ton more of stuff that you have to do in order to ultimately get to a system that you would recognize fully as alive, or get to Demeter's Terra Darwinia that's capable of Darwinian evolution. But a situation in where you have synthesized no monomers versus a situation in which you have synthesized the vast majority of them is most definitely progress in origin of life research, and most definitely progress toward getting toward a system that's alive. And so I think he's being way too hard on himself there.
Yeah, I agree.
Humble.
Yeah,
Humble. he will be, that quote will be cherry picked. It's got to watch it. Anyway.
If you think about the later stages of the chemistry, before you just before you reach a living system, the chemistry that precedes that must be occurring under conditions conducive with life, or conducive to life, because there's a transition at the very last point. So the conditions just before that point are very unlikely to have changed from the conditions just after that point. But if you go back in historical time to the point at which you're making the building blocks, is there any argument to be made? Those building blocks have to be made under conditions compatible with life, or could they in fact be made in a different way? So what people have tended to assume in the past is that the conditions which are conducive to building blocks would be compatible with living systems. And this has caused some tension, I would say, in the field. If you actually look at the way biology now makes building blocks, here's what I drew out as a minimal metabolic network you'd need to make about half the amino acids and 4 of the nucleotides. So if you want to get to the point where biology is capable of making and replicating RNA and making simple peptides, you would need to accomplish this degree of metabolism using conditions which are conducive to life, if you subscribe to the diagram I just showed you. And it's fraught with problems, absolutely fraught with problems. You try doing some of this chemistry without catalysts, no chance. You've got to stop metal ions doing bad things as well as occasionally doing good things. You've got no flux control. How do you stop all of the flux going in one direction from a branch point and not going in multiple directions that you want? You've got interference between some reactions which are reduction, some which are oxidation. You've got a lack of substrate selectivity, a lack of energy coupling, and you've got the problem that you're playing with activating intermediates, all of which would be prone to reaction with ammonia, and yet you need ammonia to introduce nitrogen into these molecules that contain nitrogen. So the chemistry take on that is that that does not work.
Sorry, I was being distracted.
That's okay.
Yep.
What he was saying there about lack of specificity, I talked about this a little bit with you last time we went over abiogenesis. There are environmental factors that can increase the specificity of the catalysis. And two environmental factors are heat and pH. And so if you have gradients in your environment with differences in heat, differences in pH, that can help to specify products. And I just wanted to throw that in there.
Okay, good.
As a network to make the building blocks you need for life. So under conditions conducive to life. So we started to consider the possibility that the conditions to make the building blocks could be completely different. Or of course they could partially overlap with conditions for life, or you could have some conditions which overlap and some that don't. We need to free our minds and be less constrained by these concepts that derive from our assessment of the way biology does things. And so we decided to take a more holistic approach. We would simply use organic synthetic chemistry to experimentally evaluate the assembly chemistry of the various subsystems. We would look to see if there was anything in the synthesis or the mechanism that related those different subsystems. We would use the results of the chemistry to start to infer something about the geochemistry. So, for example, if we found that we needed ultraviolet light, not just in one reaction, but in many reactions, that would rule out a deep sea scenario. So
Danny can
clues from the chemistry.
Sorry, I had to chime in on that one. Yeah, so with the requirements of UV light, I've heard that before from other researchers like McKellion, who focuses on his UVC model. And I don't know yet, I still have more reading to do on this, whether or not there are other avenues of electrochemistry that will provide the same result. So like that's an open question to me. But another one is that even if you were to show that UV is somehow necessary, that doesn't rule out marine-based or water-based origin of life scenarios, though it would rule out a deep sea origin. So I don't want people to confuse the idea of ruling out a deep sea origin with ruling out a hydrothermal vent origin. Hydrothermal vents occur at varying depths. They occur in lakes, they occur in the ocean. There are surficial hydrothermal vents, there are deep sea hydrothermal vents. And so don't equate in your mind that if a deep sea origin does ultimately get discredited, that therefore there's something wrong with a hydrothermal vent scenario, because those are two distinct scenarios. There's only a certain class of hydrothermal vents that are particularly deep sea. The rest of them occur elsewhere. So they're still open to investigation.
And wouldn't the first 500 million years have had a great deal of volcanic activity where we would have a lot of those vents, basically? like shallower ones?
Yeah, during the Hadean, I've commonly heard it said that hydrothermal vents are like the most frequent thing that you're going to see in the Hadean.
Right.
They're all over the place, and they're occurring in many different kinds of environments. They're occurring in marine environments where the salt content will be higher. They're occurring in freshwater environments. They're occurring, toward the surface and at the bottom of the ocean, everywhere.
Cool.
Yeah, but
Okay.
you also have to point out that his thinking is that you don't need to have the same place where you have one type of chemistry is going and to be useful for the biology in another place.
Yep, that's something that I've seen referenced in a bunch of recent articles too, is the idea of having like multiple environments and stuff like that. Another thing that I would like to stress though is that you can have one environment with a multiplicity of environmental ranges. Like there can be one environment, but that environment can host multiple different features. Like for example, to give a more concrete image of what I'm talking about, with a hydrothermal vent, and the reason why I'm going to kind of harp on this is because that's the particular model that I favor. You definitely check out the terrestrial origin models, the hot springs and all that. Those are competitors.
He will show you after a while exactly what you're talking about.
But maybe they aren't competitors. Maybe they're collaborators, because if there were a lot of these hydrothermal vents along with a lot of hot springs, there's a possibility of nicking from one system to the other, right?
That's totally true. You could have chemistry going on in hot springs, and with the Diemer-Damer hypothesis, the water from those hot springs flow from high elevations down to lower elevations. So they could make it into, for example, alkaline freshwater lakes and stuff like that, where hydrothermal systems are operating and the chemistry can continue there. But another thing about it, like to give a more concrete example of what I'm talking about with the vents, you have a lot of gradients. So you have a heat source of hotter alkaline fluid pumping up through the crust and through the vents. But then on the periphery of the vents, it's much colder because that's where it intersects with the colder ocean water. So there you don't just have like a uniform hot environment. You have a heat source that is surrounded by much colder areas. And so that's one way in which you can have an environment that actually has a bunch of different things going on, and you can't describe it in just some simple uniform fashion.
Right. Yeah. Okay. I just sent an email. I just linked our video to Bruce Damer. See if you'll watch it.
To give us some idea of the geochemistry. And as I said before, once we have an idea about the geochemistry, that's going to back inform chemistry. And then we'll toggle between the last two things and try and improve our assessment of the scenarios and try and improve the chemistry. A starting point. If you think about what biology does in terms of fixation of material, biology fixes carbon from CO2 and fixes nitrogen from N2. That is a major accomplishment. Either one of those jobs is a major challenge. And if you want to synthesize the building blocks which contain carbon and nitrogen, there's no point fixing carbon without fixing nitrogen, and vice versa. And fixing both simultaneously, de novo, seems pretty tricky if you start with carbon dioxide and nitrogen. But if you start with the molecule hydrogen cyanide contains carbon and nitrogen. The nitrogen's at the right oxidation level, pretty much for biology. The carbon is very close to the right oxidation level for use in biology. So let's not pick our building block precursor molecules, our feedstocks, on the basis of what biology now does. Let's actually just think, what would be the most useful thing to get the elements we need to actually make the building blocks that we want to see synthesized.
Dannikins.
And
sorry.
Right.
So what he's talking about there is the principle of continuity, where a lot of researchers stress that evolution tends to be conservative. It's a tinkerer. It doesn't often come up with things that are totally de novo. it works on what it previously had.
Right.
And so a lot of researchers really like to focus on trying to understand abiogenesis in terms of the way that biology operates now. And so, for example, a little later in this lecture, you're going to hear him talk quite a bit about hydrogen cyanide. It's coming up pretty soon. And one of the objections that you'll hear from researchers like Nick Lane is that today, hydrogen cyanide isn't used. And so, you know, it breaks the principle of continuity to suggest that hydrogen cyanide was used at origins. And so, you know, I think it's important, my personal take on this is that I think it's important to abide by a principle of continuity, but at the same time to recognize that if you have good reasons for letting go of that principle, thinking that at the origins there was something different going on, you shouldn't be so strictly tied to that principle that you ignore it. And that's why I'm pretty open to what Sutherland is about to say about hydrogen cyanide contra Nick Lane, because when I've looked through the literature, I've seen a lot of really, really good results coming from hydrogen cyanide. This is, again, something else that I have to look a lot more further into because I haven't read a lot of these papers, but I have come across them and their results, which are all consistently positive for what hydrogen cyanide can do. And so, you know, if it is the case that further research shows that, for example, Lane's mechanism of hydrogen and carbon dioxide doesn't really work, the results so far for that are also really positive. But just hypothetically, if it doesn't work in the future, I don't see why we should be so blindly restricted to this principle that we think we can't abandon it under certain scenarios where we have good reason to.
Yeah, there certainly wasn't any continuity in the environments. I mean, if you look at to the Hadean and now, I mean, they're certain that you wouldn't expect a continuity. I mean, it was very, very different for the first 500 million years than it is the first billion or 2 billion years. Anyway, okay, SciGuard is in our side chat about that. Hi, Sci.
Hi, Sci.
Okay, moving on.
You might look at that previous slide and say, well, hang on just a minute. You just showed us that the carbon in building blocks, the carbon is roughly the oxidation level of formaldehyde. So why actually don't you start with formaldehyde and try and start to make carbon-carbon bonds and synthesize things from formaldehyde? Well, the problem is that getting two formaldehydes to join together is very difficult. They're like two little bar magnets.
Give the sound. Mike, you pressed the wrong button.
Yeah, speed went mute. Uh-oh.
Oh, I'm
if you
sorry.
add hydrogen cyanide to it, it forms a molecule which is called a cyanohydrin.
Okay, let me back up a little bit.
The problem is that getting two formaldehydes to join together is very difficult. They're like two little bar magnets, and you're trying to get the north poles to combine. They repel each other. They don't want to combine. So you have to use a trick in chemistry called Umpelung, polarity reversal, to get this to work. You can make carbon-carbon bonds very easily from formaldehyde if you add hydrogen cyanide to it. It forms a molecule which is called a cyanohydrin, glycolonitrile from formaldehyde and hydrogen cyanide, and that's a reaction that's so easy, so efficient, that even I could do it if my group allowed me to work with hydrogen cyanide. And they don't, for good reason. And so a key finding in this area was Dougal Ritson, one of the postdocs in my group. He found that he could reduce hydrogen cyanide by one electron chemistry to the molecule on the left there, formaldehyde imine, which spontaneously hydrolyzes in situ to formaldehyde plus ammonia. So you take hydrogen cyanide, hydrogen sulfide, which is about 10 times more toxic, put it in water and irradiate it with UV light. And then when I get back from a foreign trip, conveniently timed to be away when he was doing it, I got back and discovered that he'd been able to reduce the hydrogen cyanide to formaldehyde imine. It hydrolyzes in situ, it makes formaldehyde, it makes glycolonitrile. And it then does it again. It reduces the nitrile group, hydrolyzes the amine, and makes the carbonyl compound. And then it does it again, and you make glyceraldehyde. So hydrogen cyanide, hydrogen
And glyceraldehyde is very important.
sulfide, UV light, you make the 2 and 3 carbon sugars, which taken together make a 5 carbon sugar, ribose, for RNA synthesis. And the trick here is 1 electron chemistry. Hydrogen cyanide is predisposed towards being reduced by 1 electron and a proton, either simultaneously or stepwise, because the iminar radical, drawn on the top reaction line, is a stabilized radical. So 1 electron chemistry turns out to be a very efficient way of reducing nitriles. And you can generate these electrons by photochemistry from an anion. The electrons of an anion are loosely held, hit them with a UV photon, detach them, they become hydrated in water, and then they do reduction chemistry in water. And this is high yielding chemistry. You can, if you want to optimize this for the synthesis of glycoaldehyde, you can make 60% glycoaldehyde from this. And so add a few more reactions to this, and this basic chemistry, we call it cyanosulfidic chemistry, is chemistry that takes nitriles and homologates them, makes them longer by 1 carbon at a time. And it turns out that you develop a reaction network from this that just naturally assembles about half the amino acids and precursors to the nucleotides and glycerol needed for glycerophosphate lipid synthesis.
Yeah, so right there, all 3 things from the same reaction network, right? Nucleic acids, amino acids, and basically triglycerides, or triglyceride, yeah, glycerol and fatty acids. So I mean, that, I thought that was really significant. That's very,
Yes,
very.
it is. Yes, it is. And I pointed out in one of the paper, because there was about arginine question usually. They said, oh, you cannot have arginine. It's not possible to have arginine. What kind of interaction you may have in that case with the peptide? Okay, that's it. If you have if you have nucleic acid, you must have arginine anyway.
Right. Yep. Cool.
I see some people in the chat talking about Dr. Gart and Andrew talking about some discontinuity going on in the evolutionary process. And I myself wouldn't really necessarily have a problem with that at all. I'm sure that it takes place, and the further question of like which more commonly or frequently happens, discontinuity or continuity, is something that you would have to really gauge by looking at many different examples and seeing which one ultimately wins out. But I think one last thing I want to add in there is that if you're going to go for a discontinuous model for abiogenesis, another important aspect is you need to explain how you went from something that was originally used and is no longer used to the current extant system. And I think explaining that if you're going to appeal to something that is not used in extant biology, you need to have an account of the transition in order to make it really plausible.
Based on this, also your thought, just to continue your thought about discontinuity and the principle of continuation, you have to pay attention where you put this principle of continuation, before or after the Darwinian evolution occurred. And that's very important, because mostly the principle of continuation, it's valid most of the time. That could be some exception, but most of the time are valid after, once you have Darwinian evolution. So that's it.
Yeah. Okay, proceed.
Yep.
And these molecules are made, and very little else is made alongside it. So these are just fundamentally the reaction products of hydrogen cyanide under 1 electron reduction conditions. So this suggests basically that the chemistry is not random high energy chemistry making thousands or millions of different compounds, including the ones you want. It's chemistry that's highly selective to make just the molecules you actually want. And it suggests that the conditions are to a large extent not overlapping with those conducive to life, because high energy ultraviolet radiation and so on is not good necessarily for living systems. So it lets us answer some of the questions. Are completely different chemistries needed to make the various subsystems? No. Amino acids and nucleotides are highly related when you look at the chemistry that makes them. Structurally, they look completely different. But when you look at the chemistry that makes them, the chemistry that makes them is highly related. So that suggests that amino acids and nucleotides were probably made at the same time, not distinctly, and lipid components too. Is spatial separation of parts of the synthetic network necessary? Yes, we think you couldn't operate all of this network in one pot. You have to operate it in different, probably 2 or 3 different aqueous environments, and then mix them afterwards. And I'll show you how we think that could happen in a minute. And as I said, initially at least, it looks like this is intrinsically favored chemistry, leading to a restricted set of products that just happens to work. And so for those of you with a geochemical background, a rough geochemical scenario, which we think is consistent with this, is that you can stockpile hydrogen cyanide by allowing it to react with ferrous ions in water to give ferrocyanide. Hydrogen sulfide can be stockpiled as iron sulfides. Phosphate can be accumulated because it's not precipitated as the calcium salt in the presence of carbonate. And David Catling and his group have elaborated on these ideas in the paper that's indicated that.
Remember the phosphor question.
Simply by hitting it, which is.
Did you
remember the phosphor question that we was discussing before?
Oh yeah, exactly. Yep.
Geochemically possible, either by mafic intrusion or meteorite impact or whatever. So you simply heat ferrocyanide and you get cyanide back in the dry state. And then if you dissolve it in water, depending on how much water you add, you can get any concentration of cyanide you want in quantity on an early planet. And the system we're thinking of is some sort of sloping surface where and then thermally metamorphosed, and then precipitation or ice water drainage basically creates rivulets or streams which run across that surface, dissolving materials in the sequence in which they're encountered, and at confluences between 2 streams of different histories, you get mixing of different chemistries. For example, if you have a stream that contains sodium or potassium cyanide and it flows over ground containing a metal sulfide, it will dissolve that metal sulfide and liberate hydrosulfide, the conjugate base of hydrogen sulfide. So that would be equivalent to a person in a white coat adding cyanide first and then waiting half an hour and adding hydrogen sulfide without a person with a white coat being there.
So
what about synthetic?
I think this is an opportunity for the hot springs people and the hydrothermal vets people to get together and make a truce. because I think there's an opportunity for both to have contributed in that scenario, right?
Mechanistic.
Yes, there's something related to that I wanted to add, and I'm losing the thought. Let me see here. Well, okay, actually, I'm sorry. The thought that I had was kind of unrelated, but it's really just to stress that, what he's pointing out there about how you have these sequential steps and you don't need a guy in a white lab coat there doing it. I think that that's an important response to people like James Tour and all the others who are talking about how chemists go about their job and how enormously complicated it is, noting how nature can do the same things. It all depends on the dynamics of the environment.
Yeah, the thing that kills me about James Tour is he's a synthetic chemist, right? Organic chemist. And synthetic organic chemists spend all of their time finding new pathways to make things happen, right? So that means there has to be pathways to make things happen. You just have to find them. So, I mean, that is his job, and yet he denies that it was possible. Now, isn't that interesting? Do you see the conflict there with what he does? He finds pathways. I mean, really, really complicated, strange pathways in order to create something, right? And he's just claiming that, that can't happen unless somebody's doing it. Not necessarily. If there are pathways, they can happen. So anyway, I don't know if that point lands or not.
Relatedness, just a couple of examples of this. So if you look at leucine and valine, these two amino acids in the bottom left, and you look at glycerol phosphate lipids, they don't appear to be structurally related. But notice that the chemistry of dihydroxyacetone on the left there, if you reduce dihydroxyacetone with hydrated electrons, you make a radical anion, which just naturally undergoes two different fates. One is protonation, and ultimately the production of glycerol. The other is elimination of hydroxide, which leads ultimately to the formation of acetone. And acetone, under the reductive homologation conditions, generates the two methyl groups, the geminal methyl groups of valine and leucine. So no structural relatedness, but connectedness or relatedness through their synthetic chemistry of their intermediates. Another quick example, nucleotides, cytidine, cyclic phosphate, and arginine don't look remotely similar. But both of the syntheses in this reaction network rely upon the initial equilibration of cyanamide with a carbonyl compound to give an adduct, which then delivers the cyanamide intramolecularly to a proximate nucleophile, a hydroxyl group in the uppercase, a nitrogen nucleophile in the lowercase. So highly related chemistry resulting in very disparate structures. So the connection is the synthetic relatedness. Amino acids and nucleotides are connected in that way.
That's what you were pointing out with arginine and, right, Demeter?
Yeah.
If you don't capture a cyanamide in that way, and you just start trying to homologate that amino aldehyde shown on the left, it ends up cyclizing. And you can't avoid that cyclization, and that, we think, is why you can't homologate to the point of making ornithine or lysine, because you can't avoid the cyclization, and it's why you end up making proline and not those other amino acids. So this is saying that nature didn't choose its amino acids from a huge number and just select the ones it wanted for functional reasons. It was given certain amino acids by predisposed organic chemistry, and those amino acids turned out to work.
Here I have just a little bit.
Yeah, that particular thing changed my mind on something. So,
from the perspective of what is available, he is correct. But also you have to pay attention that once you have Darwinian evolution going on, you have only function and something which is functional and makes things more stable. And then you actually have to pick up from what is available. And that's why the actual abundance of the amino acid, it's not something that determines what the code will be, what will be the first amino acid. So there is no abundance and chronology. Abundance versus chronology, there is no such a link. That's what I'm trying to say.
Okay, moving on.
And so we think the most ancient amino acids are Gly, Ala, Ser, 3, Arg, Pro, Lu, and Val, and we think those are more ancient than amino acids such as lysine and the aromatic amino acids cysteine, methionine, and so on. And we actually think that all of the subsystems that I delineated at the outset are connected and related. But the two that show the strongest connection are the amino acids and the nucleotides, or RNA and proteins. They seem to be so heavily connected that we have to consider them more in detail. We see that in modern biology, amino acids or proteins and RNA are always working in concert, or almost always working in concert. You can see from the synthetic chemistry that the amino acids and the nucleotides are made by related chemistry. So why not then go ahead and consider them as having been connected for their whole histories? And what that suggests is that we should start looking for chemistry in mixtures of nucleotides and amino acids that does something more interesting than the chemistry of either pure nucleotides or pure amino acids. So the mixtures might offer us something that we didn't get by.
Does that make you happy, Demeter?
Did you realize that up to now, he never used the term RNA world?
Yeah.
Never been written RNA world. Even this vague diagram about aliveness and so on, he didn't put on the bottom RNA world.
Yeah.
So that's very interesting. So
Yeah.
he is not RNA world guy.
Yeah.
There's in the recent debate between T-Jump and Taylor,
Oh my.
Taylor was talking about how people started off in an RNA world and now they're going to an RNA-Protein world, and she viewed that as just kind of a thing of desperation, admitting that the one hypothesis doesn't work, and so now they're desperately appealing to another. But if you actually study like the history of the development of the different ideas, you get a different picture where various researchers, because of their specializations and personal preferences about what theories they liked, investigated which components came first. But as time has gone on, it looks more and more like they're converging on an RNA peptide world rather than just abandoning the RNA world out of desperation. Like the people who are originally supporting metabolism first, and those who are supporting replication first, and those arguing first for a lipid world. As the conversation continues, more and more researchers are coming to agree with one of Sutherland's main points here, that it doesn't have to be separate. These lines of development don't need to be separate at all. And I think that that's just an advance of research rather than an appeal made out of desperation. And I think that it bears itself out when you actually study like the historical development of the conversation in the literature.
Yeah. When she said that in T-Jump's debate, I was so excited because I'm almost certain she got that from me when I was talking to her on Bread of Life's channel. So I'm pretty sure that was the first time she had heard of RNA peptide world. So
yeah, a lot of people had a lot of bad things to say about her in that debate. I only watched bits and parts of it, but I didn't think that she was terrible. She had some interesting things that she said that I didn't think T-Jump was actually really familiar with. But definitely that particular point of hers, I don't think it bears itself out when you actually read the progress of all the papers being published and the way that the thinking has developed over time.
yeah, I think she suffers from having been by herself thinking about this with herself, and she hasn't been in the community long enough to become seasoned. She will become a seasoned creationist eventually, but right now she's she just kind of came out of the box, and you have to give her a break, because I don't think she wasn't on YouTube until about a week and a half ago or 2 weeks ago. So she'll get better with her.
Dr. Gart mentions a Wacht holder. I'm not familiar with that researcher. Assuming that he doesn't mean Wachter-Schauser, I'm going to have to look that up. Wacht holder. I've never heard of that researcher before.
I'll take a look. Okay, I'm going to play.
By considering the simpler systems. And so we're starting to wonder, might there be synergy in the emergence of RNA proteins and ribonucleoproteins from their components? And this is when we started thinking about going from building block synthesis to actually trying to synthesize polymeric systems, macromolecules. And if you do that, you can't really progress without looking at the genetic code. This is, to a synthetic organic chemist, this is pure wonder. It's a beautiful relationship that exists between the language of nucleic acids and the language of peptides, determined 50, 60, 70 years ago, whenever it was. Basically, it's an amazing relationship. And I'll just point out one thing for you from the outset. There are boxes containing amino acids in the genetic code, and people distinguish two different sorts of boxes. There's a box which is fourfold degenerate, where the last base of the codon doesn't alter the nature of the amino acid. They're called family boxes or fourfold degenerate boxes. And there are other boxes which are split. They're called split boxes or twofold degenerate boxes or something similar. So let's keep the genetic code in mind when we're starting to think about how mixed nucleic acid and amino acid chemistry might play out. Let's also keep in mind how information transfer in biology operates. So the genetic code is read by the decoding center of the ribosome. Okay, so the decoding center in the 30S subunit of the prokaryotic ribosome matches tRNAs with particular anticodon sequences to the corresponding, through the Watson-Crick base pairing rules, codons on messenger RNA. And that's one part of information transfer. But the other part, which is equally important, because the whole thing wouldn't work without both working, is the way in which the right amino acid is attached to the right tRNA, or so-called cognate tRNA. What happens is that the protein, different proteins for each of the amino acids, recognizes the amino acid, and then it looks at the various tRNAs available to it and uses what's called identity determinants to specify that it's the cognate tRNA for the amino acid it's just recognized. And then it attaches the two together using ATP via a mixed anhydride intermediate. And so how did this whole system evolve is a really interesting, difficult question. But interesting, difficult questions means it's something that's exciting to start thinking about.
Dannykins.
And if I zoom in just...
Yeah.
Shortly, he's going to get into the idea of like a second genetic code and stereochemical affinity and stuff like that. So this is something that Andrew was talking about in the last big show that we did on abiogenesis and the work of Rodin and the particular paper that Andrew referenced there. So this might make Andrew happy to listen to the next few minutes. And I also I saw that Dr. Gart said that he actually did mean Wachter-Schauser. And just to say, Wachter-Schauser's a friggin' boss. There's one paper, he's got a ton of groundbreaking papers and really interesting things to say. But one of them, a short one that I really enjoyed that was pretty recent, was In Praise of Error. And he, in that paper, that's kind of a creationist dream paper. It's something that I'm sure eventually we'll see them quoting from, because he really goes in on a lot of different researchers and starts to criticize their various models and picks them apart. But he's a really hard-hitting academic. He's a lawyer, I believe, by profession, but a great example, actually. I think it was Gutsick Gibbon asking for examples of researchers that are outside the field of specialization who have nevertheless made like a fundamental contribution to the field. And one person that I would definitely recommend is Wachter-Schauser, because I'm not sure whether or not he has PhDs in the specialization. So maybe my example doesn't totally count. But I know that he's supposed to be a lawyer by trade, and yet he's a fundamental, very important researcher to look into when you're looking at abiogenesis.
One of the things coming up, I think, are this bit about two different tRNAs, or two different tRNAs attaching to the RNA, to the mRNA at the same time, right alongside each other. One of the things that have always concerned me is how it got to step along the RNA 3 codons at a time, right? And I think he suggests that there were 2, or it was going 6 at a time or something on that order. I don't know, it's coming up anyway.
Yeah, but Mike, that's it's coming. So don't go, don't jump there. Yeah.
Don't jump there. Yeah, I won't jump. But I'm excited.
It's a little bit on the anticodon loop of tRNA, and just pick out a couple of features to emphasize to you. There's the codon at the bottom with one direction, 5' to 3' right to left, and above it, 5' to 3' left to right. And what you've got is the 3 bases of the anticodon that binds the codon, And then you've got a little bit of modification that biology has made to the tRNA. On the right-hand side, the purine, that's at position 37, is often modified to assist in the decoding of AU-rich codons and anticodons. Okay. And on the left-hand side, the wobble base at position 34 is often modified to enable the decoding of the split boxes, right? So tRNAs have canonical nucleobases and they have modified nucleobases, and the modified nucleobases have those roles. The structure of the anticodon loop is maintained by a U-turn, which is a motif where a turn is induced by the binding, the hydrogen bonding of the N3 hydrogen of uridine 33 to the phosphate, internucleotide phosphate bond between residues 35 and 36. And that's what keeps the loop in the form of a loop and not collapsed. And as I said, some of the bases are modified. And biology goes to great length. 4% of the E. coli genome is devoted to enzymes that modify RNA, tRNA in particular. So biology is going to a great length to make all sorts of modifications to make the decoding more efficient. And some of these modifications, I mean, the Q base, that's not a typo. There's no nitrogen in the second, in the N7 position of the purine. That's a highly modified base. Biology is really going to great lengths to do something here. If you look at the modifications to base 37, here they are again. Notice that a lot of these modifications are not capable of base pairing. So they're introduced post-transcriptionally, but they couldn't have been introduced in a primitive genetic system by copying chemistry because they can't hydrogen bond to a Watson-Crick base pairing counterpart. So if you found they were useful at a particular position, you couldn't recapitulate that in subsequent generations by information transfer. You just have to rely upon random introduction. So what you end up with is what these characters, Crick, Brenner, Klug, and Pijnik from the lab I now work in, identified as being a notoriously difficult problem. They were not kidding, okay? The origin of protein synthesis is a notoriously difficult problem, and I like the next sentence or so because I think it actually points to a really important aspect of the problem. We don't mean the formation of random polypeptides, but the origin of synthesis of polypeptides directed, however crudely, by a nucleic acid template. And this is the crucial bit, and of such a nature that it could evolve by steps into the present genetic code. So any primitive system we're going to invoke has to be something that we could imagine could proceed by a series of steps into the system we now see in biology. We can't just invent some macromolecular system which makes peptides. If it's not capable of transitioning to what we now see, it's irrelevant to the origin of life. It's interesting chemistry, but it's not relevant to the origin of life, I would argue.
Danikins.
So now down to the meat of the chemistry.
I'm sorry to interrupt right here because this is one of the most important aspects of the lecture. But what he just said there about highlighting the transition, about making sense of the transition, that kind of ties into what I was saying about saying before, that if you're going to suggest something that breaks continuity, it's important to show how the transition is made in order to ultimately have like a plausible model, and I do think that's important. And the last thing is Icarus was saying that I'm butchering the name of Wachter-Schauser. It's possible that I am. It's just that I've always heard the name pronounced that way, and I'm getting that from the audiobook version of Nick Lane's The Vital Question, as well as a few researchers that I've heard mention his name, and it's pronounced that way by those researchers and by that audiobook narrator as Wachter-Schauser. But
you are famously wrong on this channel, so
yes, I am. So yeah, it's totally possible that that's not the correct pronunciation of his name, but those are the sources where I'm getting the pronunciation from, if that makes any difference.
Yep. So biology. Okay, Sigurd has a question here, or yeah, biology is made and implies evolution has begun. I'm not so sure about that.
Well, biology's mate is exactly mean that evolution has begun. So because if biology mean you have evolution, you have Darwinian evolution. So that's one of the things that makes the difference between chemistry and biology. Because at the moment when you have evolution, Darwinian evolution, you actually are capable to evolve. So otherwise, you're capable of something very small to start to make it more functional and actually to grow, to make it more complicated. And Sutherland, he exactly that what he pointed in the previous 2 minutes was you cannot just make some humongous structure which makes proteins. You have to start from something more simple. That's exactly what he said. However, I still want to mention that Sutherland does not have definition of exactly where the boundary is. He's just saying things which are correct, but he's not to the point that he exactly pointed out and said, okay, that is the moment where you have biology, and before that there is no it is chemistry. So and he acknowledged also that by the way, by now he's pointing clearly about the translation is the most important moment of this particular boundary, not the replication.
Okay.
Okay.
Here's my first problem with Dr. Gart. I heard him say in a stream on one of the Young Earth Creationist channels that in 70 years there's been no progress made in abiogenesis research, and I'm just wondering how in the world can he really say that? So
he's poking fun at us.
Okay, because I mean, I just if he's going to start listing his problems, I just wanted to start to list mine.
Okay, if he didn't agree with those guys a little bit, they would eat him alive. So you got to give him a break. So
yeah, we are not discussing right now. We are not making dispute or discussion.
Okay.
We have, and a lot of people have been sort of attracted by the chemistry that Leslie Orgel described to make the phospholester bond of RNA by using activated intermediates which don't require obligate enzyme catalysis, so-called idazolide intermediates. These things will react on their own to actually make phospholester bonds. But it's known, and it was Orgel that pointed this out, that they react with amino acids. At high pH, they react with amino acids to make a nitrogen phosphorus bond, to make a phosphoramidate. At lower pH, they react on the amino acid carboxylate to make a mixed anhydride. So if you're trying to do RNA replication chemistry using imidazolides and you're in the presence of amino acids, you're either going to end up making phosphoramidates or mixed anhydrides as byproducts. And the systems chemistry mantra now is, what used to be viewed as an undesirable byproduct is now an attractive possibility.
That I like. That one statement makes me happy. So.
Also, I want to mention here something that everyone should recognize. Look, in the alkali conditions, you have reaction with the phosphate actually interacts with the nitro group. However, that's not what usually happens in the biology. Usually the amino acid is carboxyl connected with the RNA. So in acidic conditions, you have the right side where carboxyl interacts with the phosphate. And guess what? The RNA by itself is stable in acidic condition, not in alkali condition.
Yeah.
It's a polymer.
Yeah, I keep hearing this bit about RNA is like totally unstable, and I'm not sure. I haven't seen that borne out in the papers. How unstable it is. I don't know.
It especially with like increases of heat, it breaks down really rapidly, which is why they keep it on cold temperature after they synthesize it and stuff. But
isn't the half-life something like 48 hours or something like that?
Well,
it gets
guys,
it can get
yeah,
it can get down to
about,
minutes.
Yeah,
It can get down to
about
minutes
this,
with high temperature.
yeah. So about the stability of RNA, there is a huge, enormous research in the 70s. So we should not even try to right now to discuss that.
Yeah.
And I'm gonna, like I know that you guys are good friends with Dr. Gart, and I don't mean to put on my boxing gloves here very much at all, but he was saying in the chat here, he says, that wasn't me, and if you want to have an argument, we could do it elsewhere. I don't know you. That was you. After the release of the Discovery Institute video, long story short, there was an after show on the Standing for Truth channel in which you specifically said that. And I'm not too concerned about how well we know each other. I'm saying that because I really took issue when you said that. And so, regardless of how well we're acquainted, I think that that's a demonstrably false statement, and I don't think that it was good that you said that. But we can take up that conversation elsewhere if you desire to.
I already yelled at him about that one, I think. I don't know. We had a conversation. Anyway, okay, we'll drop that. Okay, we're going on.
For some sort of mixed chemistry. So the first reaction I just briefly mentioned to you is the fact that we discovered that if we take a partial duplex of RNA, so two strands annealed together, one of which has an overhang sequence, we have a 5' phosphate on the short RNA strand, and we attach phosphoramidate to that by conventional synthesis. We can use the proximity that's then induced between the carboxylate group and the diol of the RNA molecule to actually form an ester. And we can then hydrolyze the phosphoramidate bond, and we can generate an ester that way. And this is one example of a phenomenon we found to be quite general, where we can get proximity across a nicked loop in RNA structures. So here's now what we think is a better example. We make a mixed anhydride now between an amino acid and the 5' phosphate. And it turns out that amino acid can be transferred directly to the diol to make an amino acyl ester derivative. Okay? We think it's possibly one of the reasons why tRNA is still made with a 5' phosphate. We think it might suggest that the 5' phosphate was vestigial, representative of early function. Now, that reaction is, we scoped it out in a limited form.
Mike, can you stop just for a second? Yep. Sorry.
Yep.
Just one step back to the same diagram.
You want to go back one?
Just a little bit to the same what? Yeah, okay. This one.
Yep.
So here I want to point out. So remember, one of the reaction with the nitro group are more prone to alkali conditions. In the other reaction, which he pointed, so for the carboxyl group, it's more like a pH 6 or something. Probably was in this slide on the previous slide.
Okay. You said you want to go back further?
Let me see the back a little bit, just one step. Here. Okay, so see pH 6 when you have the jump from one side to the other side.
Yep.
But in fact, if you have nitro group already connected with the phosphoside, I mean that's the 3, 4, 5' on the ribose. So where's the phosphoside? You assume in that case that these conditions are in more like alkali conditions. Is that correct?
I don't know.
I think it is correct. So here I have a small problem which we have to keep in mind, because if you have such a step in alkali conditions, basically the RNA is not stable too much. In pH on the right side, where you have pH less than 6 or 6, you have pretty stable RNA.
Yeah.
So that just keep in mind. That is important also eventually. Okay, keep going.
What if you have a scenario where the synthesis occurs in a more alkaline setting? For example, again, using the vents, a synthesis occurs in proximity to the alkaline fluid, but then through a mechanism, for example, like thermophoresis, which pushes away those heavier molecules toward the more acidic and colder areas of the vents where they're interacting more with the colder seawater or the colder fresh water. Would that solve your problem?
The problem is solvable. I'm not saying that it's not solvable, either by gradients, or you can have in alkali conditions only the single nucleotide interaction, and after that, the single nucleotide to go interacts with the rest of the RNA. So basically, it's not solvable, this problem, but you have to keep in mind that.
I see. That's good to hear. I also see that I've made a new best friend in chat.
Yeah. We like Sai, and Sai is a really good guy. He was in a panel with a whole bunch of people that I don't think would totally agree with him, and I think he was just soft chewing it. And he may have said some things or gone along with some things that he doesn't particularly agree with, but he is a very good guy. You ought to get to know him before you start a fight with him. Unfortunately, yeah. I think he started a fight with him. So
yeah,
don't do that on my channel.
Yeah, no, I'm notorious for that. I tend to trigger people within a few seconds of meeting them. I apologize to you guys because he is a friend of yours, but I did want to hold him to account for what he said on that program. But yeah, I mean, like, I'm sure he's a perfectly fine guy, and I don't mind discussing with him. And he said that he was hoping to discuss the science, and I'm always up for a conversation like that. But I was disturbed by what I heard him say in that stream, and I wanted to bring it up. So I don't think that there should be a problem in holding people accountable for the things that they say. But I'm going to drop it from here on out.
Yep. Okay. Thank you. Okay. So now we're back. I don't know how far back we are here, but let's go ahead.
Here's now what we think is a better example. We make a mixed anhydride now between an amino acid and the 5' phosphate. And it turns out that amino acid can be transferred directly to the diol to make an amino acyl ester derivative. Okay, we think it's possibly one of the reasons why tRNA is still made with a 5' phosphate. We think it might suggest that the 5' phosphate was vestigial, representative of early function. Now that reaction is. We scoped it out in a limited form just by synthesizing individual molecules and seeing what happened by HPLC, but it became apparent to us we had to do this in a higher output.
Okay, so this is the part that got me so excited. So coming up. Okay.
In conjunction with Andras Jeschke's group in Heidelberg, we randomized the overhang sequence to see if different overhang sequences transferred one or other amino acid better than others. And the net result of that, if we represent the results in terms of these diagrams where the size of the letter indicates the frequency at which that letter occurs in the things that we sequenced, you can see that the different amino acids we've evaluated here are all basically being transferred with similar efficiency by similar overhangs. Okay, so the changing the overhang doesn't change the nature of the amino acid that gets transferred. It's not idiosyncratic with respect to amino acids. So what we did then was we picked one of the overhang sequences that terminated in CCA, simply because modern tRNA terminates in CCA. And we picked the highest ranked one that emerged from the sequencing, which was UUCCA. And UUCCA, we fixed that, and then we randomized the 3 bases next to that in the stem. So this is a duplex region. We randomized that, and then we used a selection method to see which molecules transferred the amino acid, and then sequenced them and compared the sequences for the different amino acids. And what we found, I almost fell off my chair when we saw this. Different amino acids are transferred preferentially by different sequences, dramatically so, with the exception of leucine and valine, which you notice, G, A or G, G, A or C, G. Leucine and valine are obviously related amino acids, but otherwise there's a real big variation in the transfer of these amino acids. So we've now, this is not published, we've now basically completed the analysis of all of these individual sequences by HPLC and kinetic analysis in triplicate. And so if, for example, we take the sequence CAG in the lower strand, you can see that transfers alanine. That alanine is the amino acid that's best transferred. Change the sequence of the trinucleotide to UCU, and now proline is the one that's best transferred. Change it to GUA, and now leucine is the best one transferred. Change it to GAG, and if we do the selection basically for leucine or valine, we end up with leucine and valine being transferred best. And glycine is the only one so far where we've not been able to get glycine as the best transferred amino acid for the sequence that the sequencing suggested. But if you imagine that glycine was a more abundant amino acid than the others, what that effectively means is that you can multiply the glycine curve by, let's say, 5, at which point the glycine becomes selective for that sequence, but is still not dominating the other sequences because glycine is very poorly transferred by the other sequences. So we've collaborated now with some very good modelers in Cambridge, and we do not understand what's responsible for this selectivity, I'm afraid to say. The modeling suggests something, but we don't really understand it. And so we need to properly try and understand it. We would like to investigate it using structural biology tools, but the problem with that is that the intermediates have a very short hydrolytic lifetime. So you don't have enough time to get long NMR acquisitions, you don't have enough time to crystallize things or even get cryo-EM structures of these things. So this is a struggle, but one I think we're up to. We've now said, okay.
So to me, that's kind of like where the code is coming from. I mean, it's like the beginnings of it, right? And the only thing they didn't come up with is cysteine, which I would like for my proteins to fold and to have some sort of enzymatic activity, even if they're very small. But then cysteine is a little bit different in that it has one of the stop codons, and I don't know, maybe there's a way to recover my cysteine at some point. I don't know.
So there was that paper I referenced to you a while back, statistical conversion from ornithine to arginine. So that might be one way. Apparently, from what I remember of the article, ornithine over time, just as a matter of law of large numbers, converts to arginine. And I'm not sure where ornithine fits into what Sutherland has here. I don't think he explicitly mentions it, but it's a possibility, I guess.
Yeah, but I want cysteine. Sorry.
So, so Mike, if you just give a step back
on this video,
previous slide, previous slide,
that one
here, for example, Now, and even the previous, it's okay. So, first of all, you see these nucleotides, which the red one. So you have UUCCA. The first thing he mentioned that he used CCA because the modern tRNA is using CCA. I have no idea if you do the same experiment, for example, if you don't have CCA, you have just random some else as a sequence. Is it going to have the same effect or not?
There may be.
Also, if you see this red-colored nucleotides, which is the deterministic sequence for the transfer, this is not the actual codons of these amino acids. You have to pay attention to this. That's not the actual codons. This is a totally different sequence from the actual codons of these amino acids. So we are not talking here for the codon formation.
Okay.
We're talking here for hybridization of specific sequence which transfer a specific amino acid.
Okay. Okay. Gotcha.
All right, just let's keep going.
Okay.
But one I think we're up to. We've now said, okay, well, this system has to evolve to the system we now see. So we started off with a duplex formed from a 5 residue RNA with the amino acid attached to the end. We can do it with a tetranucleotide. If we do it with a trinucleotide, we now end up where the trinucleotide has a melting temperature, which means that the system is behaving in a dynamic fashion. And so the trinucleotide at 16 degrees is on and off the template. So if we go in with an excess of the trinucleotide, we can get very efficient transfer. And the idea now is that it should be possible to get ribozymes to both improve the selectivity and get the binding down to mononucleotides. So by the time you get to mononucleotides, that's what amino acyl tRNA synthetases do.
Okay.
The mixed anhydride.
Ich hoffe, Simon Sessel was Now, so he mentioned that you need ribozyme, right, to improve the selectivity, right?
Yep.
That's what he said.
Yep.
Okay, why?
Well,
I mean, why should be ribozyme? Let's ask this simple question. Why he said ribozyme? Of course, you have hybridization process, and you have a specific transfer of amino acid. But he said, now you need some kind of improving. So where is the ribozyme coming from? Why you need ribozyme to do that? Is it more simple if you have peptide to do that?
Yeah.
Okay, let's keep going.
Okay. And one of the things you see in ribosome evolution is the addition of a number of proteins, basically, around these RNA, right? And I don't know. There's a E there somewhere. Okay.
Of an adenosine moiety or an adenylate to transfer the amino acid with selectivity. And when we think, we think is the first evidence to support a theory that was proposed quite a few years ago, largely on the work of people who were looking at the identity determinants in tRNAs. And Paul Schimmel discovered that identity determinants could be transferred from one tRNA to another, and that was sufficient to transfer the identity of one tRNA to another. And this led to suggestions that in the acceptor stem of tRNA, there was a second genetic code which was more deterministic than the classical genetic code, which resides in the anticodon stem. And we think what we found is actually evidence for this deterministic second genetic code in the acceptor stem.
Danikins.
Okay, here,
If you look.
So he said that deterministic, so the identity of the tRNA could be switched by switching. Basically, this is the experiment where you make another RNA with hybrid with different parts of the other RNA. So basically, tRNA, I'm talking. So he has mentioned that identity could be transferred, meaning a specific sequence can make simply another RNA. And that's what he meant, basically.
Yep.
And that's and this right here is what I was referencing before, what Andrew Cumming was talking about last time. We talked about abiogenesis. He referenced that paper and talked about the research of Yaris and Rodin and others, and the whole thing about a second genetic code. We'll hear Sutherland converging on some of that stuff.
Yeah. Cool. Proceed.
Mm-hmm.
Where the identity determinants are clustered, if you look at RNA species now, they're clustered both in the anticodon loop and in the acceptor stem, in particular in the last 3 residues of the stem region of the acceptor stem. So when the earliest amino-acyl tRNA synthetases were presumably smaller than the modern ones we now see. They have to do chemistry at the acceptor stem, so it's much easier for them to recognize identity determinants at the acceptor stem than it is for them to see identity determinants in the anticodon loop. And we think those are the primordial identity determinants. But it begs the question, if there's a deterministic second genetic code in the acceptor stem, what about the assignments of the amino acids to the classical genetic code? And that's when we started thinking, okay, well, let's look at the amino acids we can make. As I've said, there are these 8 amino acids we can make, shown in red. And we were amazed when we put that onto the genetic code, and we see that those amino acids correspond to the family box codons. There's a perfect correspondence with the family box codons. So this suggests that the amino acids which are easiest to make by cyanosulfidic reduction chemistry just happen to be those amino acids which biology now has assigned to the family box codons. And that's kind of weird, so it makes you think, well, what is it about the family box codons that's special? And this was recognized in the 1970s. Ulf Lagerqvist, and actually Leslie Orgel as well, though he didn't publish it, noticed that the family boxes are characterized by having either 1 or 2 GCs in the first 2 codon positions. And in the event that there's only 1 G or C in the first 2 codon positions, the second base is a pyrimidine, right? And that was just an observation that was made in the 70s. It derived structural meaning in the 2000s when people solved the crystal structure of the ribozyme. And I'll just play you a little video of the way in which 2 tRNAs sit on messenger RNA in the ribosome. You can actually see that there is, if the purine is at the second position of the anticodon on the left there, there's an opportunity for a purine at the second position of the anticodon to form an additional hydrogen bond with the 2' hydroxyl of uridine 33. So what this means is family box codons are those codons which either have the most Watson-Crick base pair hydrogen bonds or have an extra hydrogen bond that contributes in some way to what the loop is doing. Notice also that there is the 3 bases of the mini helix that's formed by stacking anticodons, or by having anticodons bind to codons, and you have the purine at position 37 stacking on top of that to stabilize it hydrophobically. So family boxes have this extra hydrogen bond. So we wondered, does that extra hydrogen bond increase the pre-organization of the loop and stabilize codon-anticodon binding? If you have a loop that's moving, you have an entropy penalty to pay for binding it to the corresponding codon. If you prefix that by having pre-organization, you don't have to pay the same entropy penalty. So we spent some money and we've now synthesized all 64 anticodon stem loop sequences, and we've used isothermal titration calorimetry and other techniques to measure the binding of the anticodon loops to the cognate codons, either single copies of the codons or duplicate copies of the codons, for reasons that I can describe after the lecture if you want. The relationship here is not mathematically clean, but the take-home message is very clear. Family box codons are those codons which bind the corresponding, family box codons are those codons which are bound most tightly by the corresponding anticodons. Pure and simple, okay? So it looks like biology has got a deterministic second genetic code in the acceptor stem, which was what Carl Vers and other people always speculated on, a stereochemical reason for coding and amino acylation. And then the amino acids have then been assigned to the classical genetic code in the anticodon loop, effectively at random, as Francis Crick predicted in his frozen accident hypothesis. So I love this because it suggests that these 2 geniuses of time gone by were both right, when in fact they thought they were contradicting each other. So I think it's really interesting.
Okay, so
You-
Simon says Uh, okay, so I just want to point out the entire section here about the stability of GC code. Basically, what he said that anti-codon, which is in the tRNA, it's positioned in a way that it's the middle base. The middle base, it's kind of more secured in terms of hydrogen extra bonds. And the shape by this additional hydrogen bonds, it's already determined that the middle base will be the most stable one in the position. Also, he said these family boxes amino acids are basically GC rich. And if I'm able to share something here, so let me share something just for a second.
Okay.
And it will be interesting. Share screen, share screen.
Screen share makes it very, very difficult.
From the window. Just 1 second, see. Alright, so I think I can do it. It will be relatively easy. Okay. Fast, share, window. Oh my goodness. Let's see what we'll see.
Congrats to Jason Torn for a good debate last night.
All right.
Let's see. Somehow your share went away. Your share went away.
Do you see the paper here? Or not? No, you don't see it.
No. Your share
Let's try
went
one
away
more time.
for some reason.
Share.
So in that debate last night, I was freaked out by that lady's expressions. Did you watch her while they were talking? I mean, her expressions. She was rather. She had an interesting, different take on things. But
she's
her.
Yeah, sorry. She was definitely animated. I liked her style. I thought that she was actually the one really pulling her team along. I don't really like her debate partner Stewart. I think that he's another example of just like a dime a dozen con man. But I thought she was interesting. I thought some of her points were disingenuous, and I'm not going to derail too much about it here. I'm just going to give one example, which is that she acted as if theism is basically just this meaningless term that you can make to mean whatever you want, and it's entirely subjective to everybody, and so therefore anti-theists don't have a case. And I think that that's
doesn't it make her a relativist sort of to the extreme, the way she was talking? But
and makes her position just as meaningless as her opposition if she was correct. So
but she got me thinking about the the pandemic because of the amount of time she touched her face. I couldn't let it go. She had such odd mannerisms when she was listening that I couldn't focus on anything Jason was saying. Anyway, I'm sorry. Go ahead, Demeter. Can we see?
Okay. See the paper. You see the paper, right?
Yep.
Which
right.
paper
so this
is
the paper. The two guys, one is Gospodinov, another is Dimitar Konev.
Oh, those guys. Okay, those two.
Those guys. Yeah, they said universal codons enrichment for GC to AU Nucleotide Composition Reveal a chronological assignment early to late along with LUCA Formation. That's exactly what John Sutherland said. The boxes, I mean, are indeed those with GC. So look here for the GC richness, how it goes. And you'll look at the first ones. Basically, what he said, all the acids with tryptophan, exception with the tryptophan, all of the acids, leucine, valine, all that GC-rich, he was able to, meaning synthesize prebiotically.
Yeah.
Right? Guess
Yeah.
what? There's something extremely interesting here. Yeah.
You're roboting a little bit because you have a paper up. I think you're streaming it.
Yeah. So, and there are lots of arguments why that should be, and one of the arguments is exactly this particular strongest interactions with these particular codons. However, the most interesting thing in this paper is called zonal distribution of the amino acid, the classes of the amino acid synthetases. And if you see this particular picture, with that,
yep.
Class 1 amino acid synthetases are here, and all of that here. Class 2 amino acid are here. See how wonderful distribution you have of the actually assignment for the different classes of amino acid synthesis. And this is not something just can come by chance. It is amazing to see this zonal distribution because that shows you.
Oops, sorry. It went away.
Yeah.
It was
because this zonal distribution, that show you that you're in an absolutely correct track here.
Yeah.
You cannot achieve such a zonal distribution if it was not correct. This particular from left to right distribution, and if you see the particular chronological distribution, how chronologically. Okay, that's the model, but I want to do something else here. This one. If you see how chronologically the different from the different acid it comes. So you have GC rich one, and the late one is AU rich, and you all have the phylogenetics of the amino acid synthetases, which fit well. So this is the figure that is achieved. It was done by comparison of phylogenetics from one side and GC richness from the other.
Yeah.
So if someone asking when actually you have assignment to the genetic code chronologically, here is the answer of this particular figure. So I will stop sharing right now and we'll continue with the lecture.
Okay. Okay, I'll link that paper, or at least the name of it, and I think I got one of the authors anyway, but people can look it up.
The thermal titration calorimetry. We've also used biolayer interferometry, which is a very sensitive way to show the same thing. So here are two family box codons binding low micromolar levels and the corresponding split box codons, 50 to 100 times weaker binding. Okay, so this is, we think, multiple techniques are saying the same thing here. We've also shown that contrary to what's in the literature, people have looked at the binding of two tRNAs to a piece of message at the same time in the absence of the ribosome. And they did it for technical reasons using phenylalanine tRNA, which is a split box codon anticodon system. And they determined that you can't bind two tRNAs to the message without the decoding center. But if you have family box codons, you can bind.
Mike, it's not in a full screen,
So
the
what
actual
we've got here is
presentation.
biolayer interferometry data that shows the binding of one codon, anticodon pair, then the second one, then unbind the second, then unbind the first. And we do that with another pairwise combination. Same thing is true. And the reason we think that's relevant is because in the early stages of translation, you have to bring two tRNAs together. You have to bring them together and get the 3' ends of two tRNA molecules to be close to each other. And we think that binding two tRNAs to message effectively is a way of bringing two tRNAs into rough proximity. And then the peptidal transferase center can bring the 3' ends into closer proximity to enable peptidal transfer activity to take place. So this would explain why, when biology was assigning its first amino acids to anticodon loops, it would have been advantageous to assign to those anticodon loops which bind the corresponding codons tightest, because then you have a chance of making peptidal transfer work. Okay. So conclusions there, the family box codon anticodon interactions are the strongest, absent base modification. Biology has then used base modification to even up all the other codon anticodon binding so that AU-rich codon anticodon pairings can be used, and biology is then assigned later amino acids to these other codon anticodon pairings as they become available when modifications made post-transcriptionally by the earliest 8 amino acid enzymes enabled split box decoding and AU codon decoding to take place. So we think the first assignment was made randomly according to Crick's frozen extant theory, not because of side chain nucleobase interactions in the anticodon loop, but side chain nucleobase interactions were important in the acceptor stem, and it was a chemical prelude to amino acylation, first by ribozymes and then by aminoacyl tRNA synthetases in their nascent form. The first proteins would have been based upon a highly restricted alphabet. No aromatics, no sulfur-containing amino acids, no negative charged amino acids. But we think the negative charge and the aromatics could have been compensated for by binding to RNA. RNA has negative charges and it has abundant aromatic surfaces available to it. And so one of the things we're now doing is thinking about what can we do with proteins with this restricted genetic alphabet. People have investigated restricted alphabets before, but not this particular restricted alphabet, which is suggested by the chemistry. Base modifications, AU-rich codon reading, split box decoding, all look late. So the genetic code's been assigned, we think, in 2 phases. One, deterministic plus frozen X and anticodon. And then as biology gets going with proteins made from 8 amino acids, it starts to sample other amino acids by biosynthesis. It starts to realize it can read other codons, and it then adds to the genetic code and gives us what we now see. And this is something we want to go ahead further and to investigate. So that's where we're at. It's an interesting sort of point for me, because I think it sort of is beginning to confirm this idea that RNA and amino acids, or RNA and proteins, are best viewed as being a double act and not 2 separate single acts in biology. Now, I basically haven't done any of this work. The work's all been done by a very talented group of
yeah, you can skip that.
almost all.
Okay. Yep. Anyway, and then he goes to Q&A after that, so I think we'll skip that part. So, but yeah, that was a pretty cool video. That a lot of exciting things in there, basically.
So there's a few questions which everybody should think about based on this particular video. And the first question, which we
okay, hang on a second, Demeter. Excuse me for about 4 or 5 minutes, and I'll be back. If you guys can talk, I will be listening. Okay.
All right.
I just have to attend to something. Okay, go ahead.
So the first question here is how the connection between this second clone and specific amino acylation of the amino acids, it's getting linked to the actual codon, how that happened. So you can assume that he explained basically that in a way that, first of all, you have a specific transfer of the amino acylation of the specific amino acid based on a sequence which is not the codon sequence, but continuation of the same acceptor RNA might have anticodon, or let's say proto-anticodon, which is happen to be more stable with the other proto. So the first events are kind of stoichiometric statistical events. But at the moment when they stuck together because they're more stable, they made peptide bond. So the first protein, in general, this is a hybridization-dependent peptide, which I'm talking about from 2018. He just explained hybridization peptide formation. And he said that to have two tRNA next to each other, you must have GC-rich anticodon. Otherwise, they will not be so stable. That's his experiment shows. However, the question is this system is capable of Darwinian evolution, as it is explained, without any peptides? Or you must have a specific function of peptide to stabilize this process and actually to make it advantageous? I don't have answer right now of this question, but I have to think about it, because that's the important point. Where exactly the first short peptide will be incorporated in this system and where will take over, right? So that's very important. And, and if you look at that schematic, and if you might able to go in the previous figure in this particular paper that you're showing here, this one exactly. So this is the bridge peptide idea. According to what John Sutherland says, you don't need bridge peptide to have this amino acylation part.
Yeah.
However, is that enough to keep going in Darwinian evolution or not? Obviously, you have to have a positive feedback from the product, which is the short peptide, in order this particular configuration to be stable and to continue as a Darwinian evolution. Otherwise, just by itself, you have bunch of interactions, bunch of peptides, but what? So what? Without Darwinian evolution, you cannot keep going. Everything will be diffused. It will not continue. And that's a very important point. He said that they will start actually to think about what the peptides will do. But that's for his next step of research. So lots of questions. So another question is, okay, if you do the experiment without CCA, just random sequence, how you will finish with the specificity? It's relevant because how you know that exactly was chosen CCA if the same effect you can have from another type of sequence,
right?
For example. And also, did he check all the other of these prebiotic amino acids about the actual specificity of the amino acylation, or he didn't check? I mean, what about arginine? If you put arginine, is it going arginine to be specifically transferred or not? Yeah. So it's a lots of questions, which is good to have a question, but also you have to think about how the Darwinian evolution evolved based on his model and based on this model, which is shown here. I mean, here.
Yeah, there's so many combinations, though. So many combinations to check out.
So
So many possibilities.
the bottom line and the take-home lesson from all of that is, yes, there is a quite feasibility, something very small of interactions to produce peptides, hybridization-dependent peptides, which will evolve eventually in Darwinian evolution and will start to complicate. Because if you don't have stabilization process based on this information, which is the sequence amino acid, sequence amino acid, sequence amino acid, you will not go further.
Yep.
So think about that. Okay, we can wrap up. So
Okay. Yep.
Got
we'll
a-
wrap up for today.
You
got
guys
anything?
got a quick question from Andrew in the chat.
Where is Andrew? Question for Demeter.
And
Let's see.
and I see there's people that are calling it an RNA-protein world. I like the what the literature usually says RNA peptide, and I think that's more appropriate because you're probably getting proteins later on.
Yep.
But I'm gonna put it up to Demeter to answer Andrew's question if he likes.
yeah, John Sutherland used RNA protein world just to kind of make overall the entire world which is coming.
Yep.
But if you're talking for actual origin of life, it gotta be peptide, because there is no way to have protein. There is no way anything more than a few amino acids. It's by chance it's not possible to make it functional. Yeah. So the chance to have a functional protein is zero. It's not just zero. It's not useful. Even if you assume that if you make protein at once, it's not useful.
And his question for you here, he says, in an RNA peptide world hypothesis, is the fact that RNA molecules can function enzymatically still considered important?
It depends what you call enzymatically here. So the small peptides interacts with the environment just based on the amino acids which the peptide contains. And that could have some enzymatic function, but more in a chemical style of enzymatic function. However, once you have a much longer peptide, you will have motifs, and these motifs can make domains, and these domains can make catalytic centers, and then you have enzyme. In fact, enzyme in biological meaning. So the actual evolution of the proteins goes from small peptides, which just interacts based on the amino acids which have, and continue a longer way of evolution going in a longer peptide, which now can fold and make up motifs, make up domains and proteins. So the protein is characterized with folding structures and have a specific beginning and specific end.
Yeah, I'm fascinated if you add cysteine to the mix with the possibility of very small proteins, very, very small proteins being
if you read this paper here, you will see another logic of the chronological order. Okay. Because the very first assigned amino acids have more function to interaction with the RNA.
Yep.
However, as long as you accumulate or makes longer peptide, now you have folding. Now it's coming the actual amino acids which helps for this folding. So are more prone to facilitate the folding of the longer peptide. That's why your cysteine is one of the latest recruited amino acids, not the first one. Because cysteine actually, it's not just for folding. Cysteine, there is a proteases which is have a cysteine there.
Yeah.
So So cystene's coming much later. You know, I'm kind of with Speed on this one because that's a lot of the work that's been done on like iron sulfur clusters and stuff like that. A lot of that's cysteine based. So how are you getting redox chemistry and respiration going without that being present?
Yeah.
So if you see the very first, so from the approach from top to the bottom, the very first domains are those domains which interacts with the nucleotides and also interacts with the double nucleotides, which is NAT and fat. This is the redox system, which is the base for the metabolism, right? So once you have domain which is capable specifically to interact with fat, with NAT phosphate and fat, you know this. Basically, this is dinucleotide. It's a ribonucleotide, which serves as a redox system. Everybody knows that from the biochemical courses in the school, right? But to interact with those, you already have to have domain structure. Understand? So to interact specifically, let's say to pull this in different directions, because you don't have just NAT or fat as a system, a redox system, but also you have to direct this NAT and redox system in different reactions. So to direct this, you need some sort of domain structure. That's why at some point, when I'm talking here, for example, for pre-LUCA here, if you talk for pre-LUCA 1 and pre-LUCA 2, this is an extremely early moment. So basically at pre-LUCA 1, you don't have bigger peptides than just several amino acids, although the actual amino acids are already starting to assign for specific codons.
Could it be?
Yeah.
So, I'm like thinking over some of the things that you're saying there, and like other thoughts are coming to my mind. Could it also be the case, like if you're manufacturing cysteine abiotically, and if the environment is handling that for you, like I've seen a bunch of papers showing abiotic synthesis of cysteine. Could it just have been that the environment was handling that for a while, and then as the environment changes and the organisms are becoming independent of that initial environment, they then at that point incorporate cysteine into its biosynthesis in the transition to becoming independent? Would that be like one possible way of making sense of its incorporation into the code?
I understand. So if I correctly understand the question, so I will try to answer in this way. The assignment to the genetic code is not identical to the usefulness to the system immediately.
Yep.
So this is two things, because something like cysteine could be useful for the system and not to be assigned. Why? Because you have statistical incorporation of this particular amino acid into peptides because of the abundance of this particular amino acid. Meaning, if something is more abundant, just by chance, more frequently will incorporate into the peptide without being actually assigned to specific codon. Am I clear?
Yeah.
Yes.
Yeah. So that's also in this particular paper, it's very clearly pointed out.
One last thing I wanted to throw in there, you're talking about hybridization-dependent peptides. You were interested for a long time in laboratory work synthesizing them, and pointed out how you didn't have the resources to do it yourself, and you were hoping somebody else did, and then you finally came across a paper that did so. So that must have also been something that made you really happy to see. and maybe if you, I remember you sent it to me. I don't know if you sent it to Speed, but if Speed has it, or if you could send it to Speed, maybe you can include that in the description, since we kind of cover that toward the end here, and people can explore that a bit more.
Was that the Yaris papers or?
No, it's not Yaros. So basically, this is a newer paper, which, I mean, the actual talk for Sutherland, which he did it right now, this talk is continuation of prior published paper that they showed hybridization-dependent peptides, how it's accumulated. Although they don't call it hybridization-dependent peptides. Let me see, can I find it or not?
At least send me the name of it. I may have it.
Yes. So let's see, can I find it? Probably I'll be able to find it relatively quickly.
I've been renaming papers all afternoon. I had 3,000 that weren't properly named. I need to hire somebody.
I feel your pain. I've got historical Jesus books falling out of my ears.
Yeah, that's the other thing. Scott keeps recommending.
I think I found it.
You got the name of it? First author and year?
Okay, I will put it in the chat, the actual title. First author is Felix Müller, and the name of the actual paper, the title of the paper, not the name of the paper. The title is this one.
Do I have any Müllers?
It is published to 11 of May 2022. It's a very recent paper.
Müller's
Prebiotically plausible scenario of an RNA peptide world.
Oh, let's see here. Müller.
See Andrew talking in the chat there, in terms of enzymes and catalysts and stuff like that. You're getting on the same idea, Andrew, and yeah, they do have catalytic capability, and other organics do too. For example, there's lipid catalytic networks also.
Yes. I mean, the catalysis might come from different sources. You don't have catalysis just because of the advantage of such a peptide or the protein catalysis is you have direction. You can regulate very well. So,
that's a 2022 paper.
Yeah, that's a May 2022 paper.
Yeah.
Did you find
it? I did. I've got it right here. Let's see here.
I recently did a search.
Hi, Otangelo. Nice to see you. Yes, I am still I don't see what else I can be. Evolutionist.
Kitty, go away. Kitty.
I recently did a Google Scholar search for origins of life papers just restricted to 2022, and I was able to pull up like over 100 easy on my browser already. They were all important to read.
It is absolutely amazing how many there are. Like Immutable and I went through and we loaded them all, I think from 2010 on to 2020, and I think we had, I don't know, 20 or 30,000 or something that on abiogenesis. It was pretty amazing, but there's been no progress.
Yeah, if you scroll down this paper, you will see how is the formation of hybridization-dependent peptides. Although, again, they don't call it hybridization-dependent peptide. So, but because of the hybridization, you have peptide formation.
You see that little one? Well, you can't see it. I downloaded this recently. So anyway, so I do have it somewhere. I have 2 copies now because it's a free download. Anyway, guys, you want to wrap it up? My raccoons are telling me that I got to feed them.
Okay. Okay.
they're-
Bye,
Invite them on the stream.
everybody.
Huh?
Thank you for everyone that just came.
Yeah. Okay, guys. So we're going to end it.
Yep.
Goodbye, cruel world. How do I end these things?